This is lecture 23. In the last lecture, we introduced hormones. We talked a little about where they come from and went over how they're controlled, the hypothalamic pituitary, fill in the blank axes. In this lecture, we'll get a little deeper into the functions of the hormones. So these are the three topics. The hormone classifications, not all hormones are classified in the same way. Hormone secretion, not all hormones are released in the same way. And then lipolysis and glycogenolysis, how hormones initiate the breakdown of fat and glycogen. As a reminder, this is the definition of hormones. This comes from the Essentials of Strength Training and Conditioning book, the fourth edition, and it's chemical messengers or signal molecules that are synthesized, stored, and released into the blood. It's mostly the blood. We'll talk about that. Released by glands and other cells. So these are the two major classes of hormones. One is fat soluble, synthesized from cholesterol, and the other is water soluble. It's synthesized from amino acids. We'll start with the amino acid hormones. Collectively, we'll call these polypeptide. Some are amine hormones, meaning they have one amino acid. If it has more amino acids, we call it a peptide hormone. And then a bunch of amino acids, it's a protein hormone. It just depends on how many amino acids are in there. But the polypeptide hormones, this class, they're made by pretty much all cells. And they're water soluble. They have cell surface receptors, and they're very fast acting, but they're short lasting. The effect just doesn't linger very long. With steroid hormones, these are longer lasting, but they're not fast acting. They take longer to exert their action, and it's just those actions last for longer. They're made in the adrenal cortex and the gonads, the testes, and the ovaries. And so you don't make steroid hormones anywhere else, but aside from the placenta, you make them in the placenta. But aside from that, you don't make them anywhere other than the adrenal cortex and the testes and ovaries. And they're synthesized from cholesterol, so they're fat soluble, not water soluble, which means they can cross the phospholipid bilayer. So they're receptors, the steroid hormone receptors. Those are inside of the cell. They're not on the cell surface. And then eicosanoids, these are sort of a third class. These are fats, mostly derived from arachidonic acid, prostaglandins and leukotrienes. We discussed those in the last block. Those are examples of eicosanoids. And they function like hormones just locally. They act on cells in proximity to where they're formed. They'll get degraded if they try to migrate. And that's the reason, like if you do a bunch of squats, you don't wind up with sore biceps the next day. The prostaglandins aren't going to be a systemic, they're going to have an effect systemically. Um, but these also act on membrane receptors, just like polypeptide hormones would. So here's a look at the three types of hormones. Epinephrine, that's an amine hormone. It's tyrosine based. It'll act on a cell surface receptor. It's water soluble, fast acting, it's short lasting. And then there's testosterone that's made from cholesterol. It's fat soluble. Um, Intracellular receptors, they're slow to take action, but they're longer lasting. And then that last one, that's triiodothyronine. It's one of your thyroid hormones, is T3 from the hypothalamic pituitary thyroid axis. And this, it's an amine hormone. It's derived from tyrosine, but it's also made from iodine. But the thyroid hormones, T3 and T4, these ones are weird because unlike polypeptide hormones, normal, the rest of the hormones in this class, they don't have cell surface receptors. They have intracellular receptors, which likens them more to steroid hormones. That's where a steroid hormone's receptor is. But T3 and T4 aren't fat soluble like steroid hormones. So they can't just cross a cell's membrane. They need to be shuttled across it by transporters. So there's sort of a middle ground between an amine hormone and a steroid hormone. Now, steroid hormones also have secondary effects that aren't genomic. Uh, they do have interactions that are associated with plasma membranes. Not every single receptor is inside of the cell, but in general, that's where most steroid receptors live. The steroid hormones go through the cell membrane, and polypeptide hormones have to act through a signaling cascade by binding to a cell surface receptor. So here's a breakdown of protein hormones compared to steroid hormones, what they're made of, where they come from, what they're soluble in, where they act, where they exert their action, and then how quickly that action arrives and departs. Beneath each list of hormone traits is a list of some examples of those hormones, just a few, not all of them, just a few. And then again, eicosanoids exist too, and eicosanoids are fats. Steroid hormones aren't fat. Steroid hormones are not fat. They're cholesterol, and cholesterol is not fat. It's a waxy thing made by the liver that lives in cell membranes. So you get it from animal food, eggs and cheese and meat and whatever. Cholesterol doesn't have any calories. It's a sterol. It's a lipid alcohol. It has an OH, a hydroxyl group. So steroids aren't made from fat. They're made from cholesterol. Eicosanoids are made from an actual lipid. In the steroid class is where we see anabolic androgenic steroids, the performance enhancing steroids, um, both endogenous and exogenous. This is something we'll talk about in the future, but not all steroid hormones are anabolic. So, like testosterone is anabolic, but cortisone is catabolic. Um, Estrogen is both. So, there are lots of kinds of steroids. Cortisol is a glucocorticoid. Aldosterone is a mineralocorticoid for blood pressure regulation. It's not something being taken by athletes for performance enhancement. So steroid doesn't mean anabolic. It just means a hormone synthesized from cholesterol, which is not fat. So here you can see the structure of some steroid hormones, cortisol and testosterone. Notice they're just modified cholesterol. And then you can see the family tree of steroid hormones above that. The androgen lineage is DHEA, dehydroepiandrosterone, to androstenedione, to testosterone, and then it can be converted to dihydrotestosterone, although hopefully it doesn't. Uh, and if you're a guy, obviously, hopefully your testosterone doesn't get converted to estradiol by aromatase. That's the enzyme that does the conversion of testosterone to the estrogen to estradiol. And on the bottom row, you see estrone, that's E1, estradiol, that's E2. There's also E3, estriol, that isn't shown here because if you're not pregnant, it doesn't really play a role. But among pregnant folk, you can make it from E2. So in general, we have glands that release hormones. In the picture to the left, that hunk of tube, that's your duodenum. That green unripe pear, right, that's your gallbladder. It squirts bile into your intestine. And then the pancreas is the thing that's poking out, and the pancreas releases insulin. And glucagon, but from the beta cells, it releases insulin, and that's shown to the right. Insulin is just a string of amino acids, 51 amino acids, which means insulin is a protein. It's a polypeptide hormone. We'll talk about that in a minute. Um, so each hormone has uh, a specific receptor that it binds to. Um, Hormones don't exert their action unless they bind. If a hormone hasn't bound, it's not exerting its action. This is another look at the cell surface receptors. Almost all polypeptide hormones, yeah, amine, peptide, and protein hormones, their receptors are on the cell surface, whereas almost all steroid hormone receptors are inside. And um, there are specific receptors for each specific hormone, and that's what this diagram is showing. Hormone A can only bind to receptor A. Hormone B can only bind to receptor B. Hormone C can only bind to receptor C. That's how hormones and receptors work. Now, getting into the steroid hormones, this is a phospholipid bilayer. You can see the cholesterol inside of the bilayer. So steroid hormones go through that cell wall to reach the receptors inside of the cell. Then those steroid hormones will bind to a receptor that's either in the cytoplasm of the cell or in the nucleus itself. Looking at different types of hormone secretion, there's autocrine, paracrine, and endocrine. Autocrine is a cell releasing a hormone that binds to itself. Remember, polypeptide hormones, um, these bind to the cell surface. So if a cell manufactures that hormone, it won't do anything until it binds to those receptors. The cell has to release it for it to have an action, so it can bind to itself. Okay, this is a really crude analogy, but if you produce gas, right? Like intestine, like asshole gas. You can't smell it while it's in your colon. You have to release it and then it can be smelled. It's the same thing with hormones. For the hormone to do anything, the cell has to release it and then it can bind to itself, its own cell surface to exert its action. Um, a less juvenile analogy would be a doorbell, right? Autocrine signaling is a doorbell that summons a response, but you can't ring it from inside the house. You have to go outside of the house to ring it. Um, some IGF, insulin-like growth factor, it's usually called mechanogrowth factor, MGF, is released in this way. It's released by a working muscle cell, a skeletal muscle cell is exercising, and it releases it, and then it binds to itself. So that would be autocrine signaling. Some interleukins, we'll talk about that soon, also have autocrine effects. For paracrine signaling, a cell releases a hormone and it binds to a neighboring cell. It doesn't go into systemic circulation. It stays local. It's like lending eggs to your neighbor. If eggs were a hormone, that's paracrine. Prostaglandins do paracrine signaling, autocrine and paracrine both. And then there's endocrine. This is when hormones enter systemic circulation and eventually they'll reach a distant cell. Not an immediate neighbor, but they'll circulate around for a while. So endocrine hormones get shuttled through the blood, and that's most hormones you're thinking of, insulin and glucagon, leptin and ghrelin, testosterone, estrogen. Most insulin-like growth factor is released as an endocrine hormone from the liver, but again, it can also be autocrine. Then myostatin. This is a myokine, a cytokine released from muscle, a small signaling protein released from muscle. We'll talk about it soon. It's classically regarded as having autocrine actions, but it has paracrine and endocrine actions too. Okay, so let's get into some cell signaling cascades, starting with glucose uptake, very superficially for now. So this is done in a couple of ways. We'll talk about that later. But insulin is how you're doing it at rest. And these are two different visuals showing the same thing. Um, but they show different stuff in the signaling. So each of them starts with insulin. It's a peptide hormone, binds to a cell surface receptor. Then a signaling cascade is initiated, which ends with GLUT4 translocation. GLUT4, a glucose transporter, being translocated, moving to the cell surface to bring sugar into the cell. That's insulin's major action is let's move sugar into the cell. GLUT4 translocation. Um, but the signaling molecule shown is like between the two diagrams, sort of funny because on the left, it shows everything but PI3K, phosphatidylinositol 3 kinase. We'll talk a lot about that later. Um, that's what converts PIP2 into PIP3, which are both shown, but you don't see PI3K there. In the diagram to the right, all you see is PI3K, nothing else. But insulin signaling, this is something we'll talk about more and more moving forward in this block. Now, if you're going to mobilize carbohydrate, not take it into the cell, but you're going to get it out of the cell, you have to break up glycogen. And this is pKa signaling, which you should remember from, at least it should be recognizable from the nociceptors. We saw pKa signaling inside of nerves too. But now we're looking at the liver or the muscle and how glycogen gets broken down, stored carbohydrates get broken up. So epinephrine, that's an amine hormone, or glucagon, that's a peptide hormone. They'll bind to their receptor. They each have their own receptor, own distinct receptors. It looks like they're the same here, but they're not. Um, and that will activate, both of them when they bind, will activate adenylate cyclase. And that converts ATP into cyclic AMP. Cyclic AMP binds to pKa, which activates it. And then once pKa, protein kinase A, is active, it phosphorylates stuff. It attaches phosphates to things. That's what kinases do. They attach phosphates to things. And here, pKa is phosphorylating stuff that mobilizes carbohydrate. At the end, you see glycogen phosphorylase. If we're talking about muscle, this is also called myophosphorylase. It's what chops up an already phosphorylated glucose chunk. It chops that off of your glycogen. And so what's interesting about muscle glycogen phosphorylase or myophosphorylase is if there's a mutation here, this is called McArdle disease, your lactate levels won't increase during exercise. And so if you have a myophosphorylase mutation and you exercise, you don't get a buildup of lactate. And so you don't release growth hormone with exercise, which tells you that lactate is a primary trigger for growth hormone secretion. Um, but this is how we mobilize carbohydrate during exercise. It's glycogen phosphorylase through pKa signaling. And then let's get into lipolysis now. This is also pKa signaling. It's the same signaling cascade, but we're in fat now. So lipolysis, lipid lysis, lipolysis. We're going to break up a triglyceride or triacylglycerol. So epinephrine or norepinephrine will bind to a beta adrenergic receptor on the surface of a fat cell. And then the same process of cell signaling happens. Adenylate cyclase is activated. In this diagram, it's called adenylyl cyclase. That's the same thing. So adenylate cyclase gets activated. ATP gets converted into cyclic AMP. That binds to pKa, and pKa phosphorylates stuff. One of the things pKa phosphorylates is perilipin. That's the coat on the lipid, which has a ton of triglycerides in it. And in order for the lipolytic enzymes to get access to those triglycerides, you need to open up that coat. So pKa phosphorylates perilipin and the coat opens. Then once the coat is open, pKa also phosphorylates hormone-sensitive lipase. You see the exact same signaling cascade here. pKa signaling, beta adrenergic receptor, adenylyl cyclase, cyclic AMP, pKa, and then pKa phosphorylating stuff. TAG, that's a triglyceride or a triacylglycerol. DAG, that's a diacylglycerol or a diglyceride. And then MAG, that's a monoacylglycerol. Hormone-sensitive lipase really works on the DAG, on the diacylglycerol, much more than the TAG, the triglyceride. Um, what does most of the work on the triglyceride is adipose triglyceride lipase, and I'll talk about that in just a minute. But first, if you see how epinephrine turns on pKa signaling, which mobilizes fat, but then also insulin is inhibiting this cascade. We'll talk about the inhibition first. So here we see beta adrenergic signaling getting started. Epinephrine is binding, adenylyl cyclase or adenylyl cyclase gets activated. It converts ATP into cyclic AMP, and cyclic AMP would then go on to bind to pKa, which would phosphorylate perilipin and hormone-sensitive lipase, but phosphodiesterase, PDE. Technically, it's PDE3B. That's the fat kind. But phosphodiesterase, that converts cyclic AMP into AMP. And AMP does not bind to pKa. So pyrrolipin and hormone-sensitive lipase are not phosphorylated. So fat is not mobilized. Um, Insulin turns on phosphodiesterase, which means insulin blocks lipolysis. And since the burning of fat begins with lipolysis, you can't burn fat in the presence of insulin because it activates PDE. Here's what AMP looks like and what cyclic AMP looks like. You don't need to know their chemical structures. It's just to show that the chemical structure is almost the same, but one of these activates pKa and the other doesn't. Here's a study where they talk about the mechanism. Downstream from insulin activation, you get PKB activation. PKB is also called AKT. You'll see it both ways. And then PKB phosphorylates PDE, phosphodiesterase, activating it. But also in this study, they inactivated PKB, i.e. AKT, and tested the effect of insulin on lipolysis. And the um, inactivated PKB blocked the effect of insulin on GLUT4 translocation. So insulin no longer brought carbs into the cell. So bringing carbs into the cell requires PKB, but it had no effect on insulin's inhibition of lipolysis, which means PI3K, which is upstream from PKB, is also activating PDE. Wortmannin, we'll talk more about that later. That's a PI3K inhibitor. And if they introduce wortmannin, you'll obstruct insulin's effect on lipolysis because it knocks out both PI3K and PKB, and both of those will activate PDE. All right, so let's look at this diagram. There's two different diagrams here. Um, in the top one, you can see insulin binding to its receptor, IRS. That's insulin receptor substrate. It passes a memo along to PI3K. The insulin is bound, and then PI3K permits the activation of PKB or AKT, as is written in the top diagram. And both PI3K and PKB contribute to PDE activation. In the lower diagram, you can see the interaction between beta adrenergic signaling and insulin signaling. On the right, that's beta adrenergic signaling. The AC is adenylate cyclase. It converts ATP into cyclic AMP. That's activating pKa. pKa would be phosphorylating both pyrrolipin and hormone-sensitive lipase. But on the left, insulin binds to its receptor and PKB gets activated. This diagram doesn't show PI3K, but that's there in real life. And so PKB and PI3K will both activate PDE3B, right? That's the fast cell version. And that converts cyclic AMP into AMP, and AMP doesn't bind to pKa. So pKa doesn't get activated. So pyrrolipin and hormone-sensitive lipase don't get phosphorylated, so fat is mobilized. So that's insulin's short-term inhibition on fat metabolism. Let's look at long-term inhibition. Um, Hormone-sensitive lipase, that gets all the praise as it like the lipolytic enzyme, the one doing all your lipolysis. But ATGL, adipose triglyceride lipase, that does most of the first lysing on the triglyceride, the tag, the triacylglycerol. Adipose triglyceride lipase does that initial lysing. And it gets activated by a protein with a very unmemorable name of comparative gene identification 58, but CGI 58. Under normal conditions, CGI 58 doesn't have access to ATGL, to adipose triglyceride lipase. Pyrrolipin blocks its access. And then pKa phosphorylates pyrrolipin, and then CGI 58 get to work. In animal models, when you knock out hormone-sensitive lipase, you see an accumulation of diglycerides, of diacylglycerol. And so that means you're still chopping up the triglycerides, the TAGs, the triacylglycerols, but the assembly line loses its DAG workers, its diacylglycerol workers, so it gets clogged up there. So pKa increases adipose triglyceride lipase phosphorylation, but only about twofold. It increases hormone-sensitive lipase phosphorylation 26-fold. And then hormone sensitive lipase can lyse triglycerides um, and triglycerides both, but it has 10 times the activity with the diglycerides. So PKA signaling really gets the um, triglyceride exposed by phosphorylating the perilipin, and then it gets hormone sensitive lipase activity going. But adipose triglyceride lipase is doing the bulk of the triglyceride lysing. Now, the expression, the genetic expression of adipose triglyceride lipase, meaning how much of the enzyme you're making, is influenced by nutrition and insulin. So, insulin seems to affect long-term lipolysis by affecting genetic expression of adipose triglyceride lipase. Looks like this. The greater the dose of insulin, the lower the expression of ATGL, adipose triglyceride lipase. So this is one explanation for how a sugary diet, a high carbohydrate diet can affect body composition in the long term. Now, looking beyond insulin at the regulation of glycolysis, um, here's an image of a fat cell. And on the left, you see catecholamines, epinephrine, and norepinephrine. Um, Epi is hormone, that's the hormone version, and then norepi, that's a neurotransmitter version. Both of these will bind to beta adrenergic receptors and stimulate lipolysis. Fat is innervated, it does have nerve connections, and fat is responsive to hormones. So epi and norepi both. So that's adenylyl cyclase signaling, as you can see in the chart in the figure here. And there's also atrial natriuretic peptide, top left in the diagram. That's released from the atria, right from the heart. And we're not going to talk about CGMP, cyclic guanine monophosphate, and then PKG. But the downstream effect of this duplicates the effect of the catecholamine. So in addition to PKA, phosphorylating hormone sensitive lipase and perilipin, PKG can do it too. And then there's also insulin at the bottom, and you know how that works. So here's a narrative explanation of how atrial natriuretic peptide exerts its function, its action. You don't need to know the details, just know that it contributes to lipolysis. It enhances lipolysis. And then growth hormone also enhances lipolysis in some slightly more circuitous ways. Um, so adipose triglyceride lipase, the ATGL, that one that does the first lysing, that's inhibited by a couple of proteins. One of them is FSP27, and growth hormone downregulates that protein. And then growth hormone also activates hormone sensitive lipase and increases the expression of it. But that would be blocked if you get a big insulin dose at the same time. Cortisol also enhances lipolysis. It binds to glucocorticoid receptors within the cell. It's a steroid hormone, so it goes inside of the cell. And when it binds, it activates the transcription of a few things that are in red there, and they code for lipolytic enzymes. But it also facilitates the release of ANGPTL4, angiopoietin-like 4. Sometimes it's called fasting-induced adipose factor because it's activated by cortisol during fasting. But it's a protein that, once it binds to its cell surface receptor, increases the amount of intracellular um, cyclic AMP, and cortisol is going to increase that. So cortisol is going to increase cyclic AMP that way. That doesn't mean you're going to get lean with cortisol because it also stimulates lipoprotein lipase activity. So you just end up storing the fat elsewhere. You do enhance lipolysis, but really what you're doing is taking fat from the limbs, right, from sort of distal fat deposits and relocating them to the viscera. So let's take some arm and leg fat and throw it in the abdomen. And then lastly, there's TNF, tumor necrosis factor. This is a cytokine with a ton of functions, but among them, it does increase lipolysis. It takes some time for that to set in. It takes hours, and so it's not fast in the way that epinephrine and norepinephrine immediately get fat mobilized, get lipids on the march. This takes time. And so transcriptional effects are likely to be the major mechanism through which tumor necrosis factor is going to enhance lipolysis. But one more look at insulin. Lots of things increase the rate of lipolysis. They stimulate lipolysis. They increase cyclic AMP. But insulin does the opposite. Insulin reduces cyclic AMP by activating PDE, phosphodiesterase. And insulin blocks the expression of adipose triglyceride lipase. So insulin blocks your short term, your immediate fat metabolism, and it blocks long term fat catabolism. So insulin is the biggest enemy of fat catabolism, both short term and long term. Insulin does other stuff too. A lot of it is very useful. We'll talk about that stuff in future lectures. Insulin isn't just an enemy of fat, it has other functions too. But for now, here are the questions for this lecture, and I will see you in the next one.